Firstly, I want to thank everybody for joining us this afternoon. And what I'd like to do would be just to introduce what's going to be happening this afternoon. And we want to talk a little bit about innovative therapies. And then Craig Campbell is going to talk about exon skipping. And then Dr. Mendell is going to talk about gene therapy. So I'm really excited to have both of these individuals here with us today. And I'm just going to share my screen if I can. Let's just share this. So it's really exciting what we see happening in new therapies and really changing our understanding of what's happening in Duchenne muscular dystrophy. And this has been happening really for very many years, in that the dystrophin gene was identified in the 1980s. And it has really led to an explosion of both our understanding and of looking at different aspects of potential treatments. It is really challenging to be faced by such a huge gene, and that has made life very difficult when it comes to looking at gene therapy. And Dr. Mendell is going to be obviously touching on that later. But it really appears that the root of the problem is certainly leading to the root of the solutions. And I think the more that we've been in this situation of dealing with children and boys and now young men with Duchenne muscular dystrophy, that we understand more and more that as we look at the science and as we understand more about both the gene itself and about dystrophin expression and about the lack of dystrophin expression and what that does in terms of the destruction of the muscle fibers and all the downstream effects from that, that each area leads to the possibility of some solutions being able to occur. And sometimes you are faced by failure, and that leads you in a different direction. But even those failures lead to other possibilities. And as we look at the different dystrophin changes, and we've looked at the different nonsense mutations and PTC and atalurin, and as we look at other deletions and where that might lead us, and we've looked to exon skipping and also to mini and subsequently microdystrophin, and looking at AAV vectors and looking at exosomes and all of these as possible cures and certainly possible treatments that may need to occur in combination to help the lives of our boys with DMD. And all in trying to allow that dystrophin that we can see so nicely in the sarcolemmal membranes of these muscle biopsies to be able to try to restore that. So I am really happy to be able to introduce Dr. Campbell to everybody. I'm just going to stop sharing my screen now. And I want to be able to introduce Craig, and we've known each other for a long time, and it's been an amazing journey as we've gone through the collaboration and increased communication across Canada with the development of the Canadian Pediatric Neuromuscular Group and the Canadian Neuromuscular Disease Registry, and now NMD4C. And Craig is the Deputy Chair of Pediatrics and Research and the head of the Division of Pediatric Neurology and the Medical Director of the Multidisciplinary Clinic based at Thames Valley Children's Centre and the Pediatric Neurophysiology Lab at Children's Hospital London Health Sciences Centre. He's an Associate Professor in Pediatrics, Clinical Neurological Sciences and Epidemiology at Western, and a scientist in the Children's Health Research Institute. His training in pediatrics was in neurology, pediatrics and epidemiology at University of Ottawa, and he is collaboratively involved in many academic and industry initiated clinical trial activities, and is also a member of the Muscle Study Group, the World Muscle Society, and is co-chair of the Treat NMD Registry Oversight Committee and Task Force. And he was also awarded twice the George Kapati Award Researcher by Muscular Dystrophy Canada, and is now part of NMD4C and is implementing innovations for the Canadian Neuromuscular Disease Registry that facilitate data sharing and patient engagement, and is also developing a research curriculum and a clinical trial network. And I would happily hand over to Craig, and thank you so much for being with us today.
Thanks. Thank you so much, Kathy, for that warm introduction. Really appreciate being here. I thank Jesse's Journey for inviting me to be here, and certainly sharing the stage with Kathy is wonderful. Similarly, getting a chance to speak alongside Jerry Mendell is a career highlight. And I've now had the opportunity to do that twice at Jesse's Journey format. So I consider myself very fortunate to have had that opportunity. And again, thank you to Jerry for spending a bit of his time with us today as well. So hopefully those slides are coming up. Just going to move into presentation mode there. Hopefully everyone can see that and hear me okay. So I am going to talk about exon skipping for the next few minutes. And then I look forward to a coffee session where I'm sure there'll be a lively discussion about exon skipping, because this continues to be exciting, but kind of almost sadly a little bit elusive innovation for DMD that I think we all wish could be upon us in a more fulsome way. But I'm going to talk about a little bit about what some of the challenges are for exon skipping. Before I do that, though, I'd just like to point out disclosures that I have. As Kathy mentioned, I am involved in a number of industry clinical trials and serve advisory functions and data safety monitoring committee memberships. And for these functions, I've been lucky to be able to not have to use any of these funds for salary support or for my personal self. And so I donate all money that I get from these functions to charity. Today, I'd like to talk about three areas of exon skipping. First of all, I'll just back up and do some exon skipping explained for people who maybe aren't so familiar with what exon skipping is. Then I'm going to talk just a bit more broadly about where we are in the global scheme with exon skipping. And then the last slide or two will be about the current state of exon skipping here in Canada. So exon skipping explained. To understand the story of exon skipping, we have to start with understanding the DMD gene. And hopefully you'll see my pointer here. This series of numbers represents the DMD gene. This DMD is one of the largest genes in humans, and it consists of these 79 parts. And you'll see they almost fit together somewhat like a puzzle piece, in that the bits of coding region that tell the body how to make dystrophin fit together in a very specific way, from the very first building block all the way to the last building block. And we refer to this puzzle piece arrangement as the reading frame. And you may have heard of this as you read about exon skipping, that we talk about, certain exon mutations or exon deletions disrupting the reading frame, or some of them don't. They maintain the reading frame. And what that means is that if you're missing an exon, if the puzzle pieces still go together, then you can maintain the reading frame. And if you maintain the reading frame, then you can imagine your body's able to still produce dystrophin, but with pieces missing. And that tends to lead to a more mild muscle weakness, or something we term Becker's muscular dystrophy. But if you have mutations that disrupt the reading frame, meaning that if an exon is missing, the two parts can't go together, then in fact the cell machinery basically stops doing what it's supposed to do, and a non-functional dystrophin protein is made, and that leads to more significant weakness in the person affected, typically by Duchenne muscular dystrophy. So another little biology step here, just to understand exon skipping, is how we make proteins like dystrophin from our DNA, from our genes. And basically there's one step in between that that we often don't think about, but is particularly important for actually not just exon skipping, but some of the other therapies geared to DMD. And that's this process of transcription, where the DNA is actually made into a sort of a biological copy of it called messenger RNA. That messenger RNA is actually the template then for the cell machinery, what we call the ribosomes, producing dystrophin. And so we actually have a step in between here that with this mRNA that we can try and manipulate. And in fact, that's where exon skipping comes in. So you might hear exon skipping being referred to as a genetic-based therapy rather than necessarily gene therapy, which Dr. Mendell is going to talk to you about. And I like this distinction because, again, what's happening with exon skipping is happening at this intermediate step on the mRNA or the messenger RNA before it makes dystrophin. And so here I've given an example just to show you exactly what we're talking about here. On this left-hand panel, you can see that there's a small segment of a dystrophin gene. I haven't shown the whole 79 exons. And in this case, you might have a boy who's missing exon 52. So that's why it's in black here. And you can see that
Now it looks like we've got a bit of a problem here. I wonder if
to do is a little bit of nucleotide molecule here that basically the cell machinery and causes it to basically skip over or miss 51. And then you can see what happens is that exon 50 and 53 actually do go together. These puzzle pieces fit, and the cell machinery can continue on to finish what it started, but in fact make a dystrophin protein that is missing a little bit in the middle. And strangely, people say, I've often heard people say, well, so you're almost making it worse to make it better. And that's in fact the case, because we know some of the most important functions of the dystrophin gene is to have the beginning and the end. And so sometimes if you're missing bits in the middle, that's actually less important, even large segments sometimes in the middle, than having the beginning and the end piece. And again, I'm talking in generalizations because there certainly are lots of things that happen in the middle of the dystrophin protein that are important as well. So basically, this is how exon skipping is working. And what are those little biochemical nucleotide molecules that we're using to trick the DNA or to trick the messenger RNA? Those are called antisense oligonucleotides. And this is a new class of biologic therapies that's really emerging. And in the last couple of years, there's been 11 new antisense products approved in the US or European Union. And in fact, most of those, as I've listed here, or a substantial chunk of those, are actually in Duchenne muscular dystrophy or another childhood disorder, spinal muscular atrophy. And so, for our community, neuromuscular community, and for DMD, you're really leading the way in understanding how antisense oligonucleotides are used in clinical practice. However, there's a long way to go still. And we have a lot to do in terms of trying to optimize how well these antisense work in people. I've shown here on the right-hand side a little chemistry lesson, but it's certainly not for anyone to necessarily take home, but only to remind us that what we're trying to do when people build these antisense oligonucleotides is make them look similar to DNA and RNA, because that's what they're trying to trick or manipulate. But you can see there's many different kinds of antisense oligonucleotides, and we, different companies, different academic labs are all trying to optimize these oligonucleotides so that they work the best to exon skip. And so not only is the chemistry under constant evolution, and you'll hear about next generation or 2.0 exon skipping, but also we need to continue to work on trying to understand optimization of these products through how we deliver them to the body, the dosing that's used, the frequency of the dosing, and even which exons are more efficiently skipped than others. So yes, this is a mutation-specific therapy, and that's important to remember because not everyone has mutations that would be amenable to exon skipping, at least not in the clinical practice. Right now, there are a lot of focus on exon 51 skipping, exon 53, and exon 45. And that's because those are the most common mutations in DMD. In fact, if you look at those three exons, you're probably up close to about 30% of boys with DMD could be helped with those therapies. But theoretically, and this is even a bit conservative, probably about 80% of boys could be helped with exon skipping therapies. If you include some other kind of less traditional ways of looking at it, certainly probably more. And that could be through using single oligonucleotides, antisense oligonucleotides, as in the demonstration I showed you. Or maybe one could even use combinations to skip several exons to restore the reading frame. Again, we need to continue to think about optimizing antisense therapy, and that includes how we study those drugs in clinical trials, because you can imagine that after some of the more common mutations, we might be getting down to mutations in the DMD gene that affect maybe only 1% or 2% of boys with DMD. And you can imagine it's very hard to run a clinical trial with such a rare mutation. And so we also have to be working with our regulators to understand how they approve therapies for exon skipping, so that we're not held to that very traditional or conservative marker of a big phase 3 clinical trial. So these are still some of the challenges ahead. So where are we, worldwide or globally in terms of exon skipping? There's currently three exon skipping products approved in the US. 2 are with Sarepta products, eteplirsen, who people have likely heard of, and golodirsen for exon 51 and 53 respectively. And then also NS Pharma has an exon skipping product for 53, which has gotten recent approval in the US. And sorry, I used the trade name here, Viltepso, which I actually don't usually do. I'm it's Vitalarsen is the generic name for that. Apologize for that. It's actually not part of my disclosures. I've not done any work with NS Pharma. There's some other companies that are in the pipeline for making antisense, Daiichi Sankyo and BioMarin are both in early clinical phase trials, as far as I know. People may remember BioMarin as being involved in the early drisapersen trials, which obviously have now been stopped. But always looked forward to what they had as a potential follow-on product. The WAVE study is also an unfortunate story of disappointment for an exon skipping product, and that was a recently stopped study due to lack of dystrophin expression and some limitation in dose escalation due to side effects in boys who are participating in that clinical trial, including at our site in London. The one thing to remember, though, for even for the products that have achieved approval in the US, is that there are post-marketing commitments for these, and most of those include completing large phase 3 studies. The approvals for these drugs were made on demonstration of dystrophin production, and so we need to still see clinical efficacy demonstrated, or basically we need to see effectiveness in some of the motor milestones and other measures we use when thinking about improving function and quality of life in boys with DMD. So what's happening from the Canadian perspective? I wanted to just point out a few things currently. So there are studies running in Canada, and what I've done here, just for ease of kind of thinking about these, is I've listed the exon skipping on the left-hand side, and then the study and the different phase and the chemistry. Down below, it would be hard to talk about exon skipping in Canada without mentioning Toshi Yokota, who's in Edmonton at the University of Alberta, who is really doing a lot of exciting preclinical work and is really a worldwide expert in exon skipping on the basic science side of things. And so he's an important treasure for us here in Canada. So there is a study ongoing for exon 53 and 45 skipping called the EXON study. That's enrolling in these centers across Canada. And those are in phase 3 studies with a PMO background from Sarepta. And then there's a number of opportunities or studies for exon 51 skipping, including a high-dose eteplirsen study, which is running in London and Ottawa, and that's a phase 3 study. There's a study of Vitalarsen, a phase 3 study that's being done in Calgary. We just completed a phase 1 study for the new PPMO, Sarepta PPMO, here in London, and we're now on to a phase 2 study of that product, PPMO. So this is a peptide, conjugated peptide PMO. So it's an improvement in tissue penetration from Sarepta. So this is their next generation of antisense. And so we're excited about being involved in these studies here in London. However, so I mentioned here the ones that are still recruiting are the 53, the EXON study, and then these 3 studies that I've pointed out here. And these are critical that we get these studies completed, because at present, we need those kinds of large clinical trials to move towards approval in Canada. And right now, unfortunately, we have no clinical access to any of the exon skipping products in Canada. So it's only through clinical trials. And I say no clinical access, but in fact, there is managed access plans through Sarepta, but of course, those are not compassionate release programs, and the drug cost still needs to be dealt with. So to my knowledge, no one has tested that pathway yet in Canada of going through the managed access route due to the high drug costs. And unfortunately, at this point in time, Sarepta has made a company-wide decision that they're not participating in a compassionate release program. So I feel the bad news, and I hate to end on this for the Canadian audience, is that there are still probably several years to go before we will have clinical access. And the reason I say that is that we will still have to complete some of these phase 3 clinical trials that are still ongoing globally, and then have that submitted to Health Canada for review. And so, so we continue to work actively to participate and finish those clinical trials here in Canada. And with that, I'd like to close. I'm happy, looking forward to taking questions in the coffee break, and I'll stop sharing my screen so we can hear from Dr. Mendell.
Thank you very much, Dr. Campbell, for giving such a comprehensive overview of exon skipping and what is available in Canada. And if people can keep the questions so that they'll be able to go to the question and answers later on this morning. And then I'm really delighted to be able to introduce Dr. Mendell, really who needs no introduction whatsoever. And when Rick Moss had spoken to me about who would you like to be able to come to the Jesse's Journey meeting, I never thought we'd be able to do this, but it was like, oh, we've got to get Dr. Mendell. I said, no problem, we'll do it. So I'm really, really happy to have you join us today. That's fantastic. And as we know, Dr. Mendell is an attending neurologist at Nationwide Children's. He holds academic positions as Professor of Pediatrics and Professor of Neurology, and holds the Curran Peters Chair of Pediatric Research. He's a clinician scientist and has got really decades of experience and a career that began with training in neuromuscular diseases at the National Institute of Health, directing laboratory projects that could be carried to the bedside. And he's really been instrumental in the true understanding of translational research and clinical translation. He's published very many articles with a definite focus on neuromuscular disease. He's authored books on muscle diseases and peripheral nervous disorders, and more recently on gene therapy for muscle disease. He was a principal investigator on the prednisone clinical trial in DMD that had profoundly influenced clinical care. And he was the PI on the exon skipping trial using eteplirsen, and now the FDA has approved that clinical use in the States. He was also the principal investigator on the first gene therapy trials for DMD and limb-girdle, and also on the SMA gene therapy trial that was approved for infants in 2017 with Spinraza. The first results of the first systemic gene therapy trial for DMD is now moving to phase 3, and I'm really happy to be able to have Dr. Mendell here today to talk about gene therapy and DMD. Thank you very much indeed.
Thank you for your kind introduction. Let me see if I can pull up my shared screen here. There we go. I think. That should do it. All right, there we go. So let me put this on. So gene therapy now has provided incredible hope for neuromuscular diseases that go from DMD, which we'll focus on today, for SMA and for limb-girdle dystrophy. And I thought I would just give you a brief introduction related to the objectives of our discussion today. Really, this centers around the most common questions that we have to address in when we talk to patients about muscular dystrophy gene therapy. One we get often asked is, where do we get the gene for testing? How does it get to muscle? Is gene therapy safe? That's a common question and one of concern often for patients. How many times does it have to be given? And finally, what happens if it stops working? So let's take an overview of how this works. And one of the things I like to emphasize in discussing really any clinical trial, but gene therapy in particular, is before we do any clinical trial, our laboratory studies have to predict a favorable outcome. We often get asked, why is Duchenne dystrophy targeted for gene therapy? And basically, when we started looking at which diseases we should try to overcome, Duchenne is the most important disease because of its incidence at 1 in 5,000. And as we move forward in the clinical trials, this incidence can justify its cost. And we have preclinical studies that show benefit and safety using microdystrophin, and I'll explain that term in a minute. And it fulfills criteria for what we call a cost-benefit ratio. That is, we have such good preclinical data that we can predict a clinical outcome. And that seems to be evolving as we get there now. So the next question I wanted to address is, how do we deliver the gene? Prior to 1999, when I did my first gene therapy trial, 2 viruses for transferring the gene were most commonly used. One is a retrovirus like HIV, and the other was adenovirus. The reason these 2 viruses were chosen is because the Duchenne gene, as Craig just discussed, is so large that we wanted to get the entire gene into the virus to use as a delivery mechanism. But things in the evolution of trials changed that significantly. And both retroviruses and adenoviruses were burdened with serious complications. Retroviruses actually insert into the way or into the genome and cause problems. A method or a complication called insertional mutagenesis. And in using retrovirus, leukemia was one of the side effects. Adenovirus, on the other hand, was a beautiful virus. In 1999, that was my first plan to use adenovirus for gene delivery. And there was a death that was related to the use of adenovirus because of a massive immune response. And that's a very well-known case called the Jesse Gelsinger case. So that ushered in the era, which for the last 20 years now, that has encouraged us to use adeno-associated virus. And despite the similarity in terms, adeno-associated virus and adenovirus are very, very different. Adeno-associated virus has a very small packaging capacity. It can't hold the whole Duchenne gene. So we'll talk about how we accommodate that in a minute. But the first step for any viral gene therapy is to remove the viral genes so that we have whatever capacity is left to put in the muscular dystrophy gene. There are 2 genes in adeno-associated virus, the rep and cap gene, and we basically splice these out or dissect these out, and then we put in what we call the recombinant microdystrophin gene. What is the microdystrophin gene? Well, if you look down here on the bottom, this is a picture of what the entire dystrophin looks like, and it's way too big. It's 14 kb or 14,000 base pairs. We have to make this, the adeno-associated virus can only accommodate 5 kb. So we have to make this much smaller. And when we make it much smaller, the next thing we do is tested in the lab. We take the recombinant now microdystrophin gene. This is essentially a DNA molecule. We put it into the adeno-associated virus that now has removed its own genes. We then put it in a syringe and then test it in mice. And essentially what we're doing is we inject it into muscle, it goes to the nucleus, then forms a double-stranded DNA that produces microdystrophin. We inject that into the mouse, and if it works, then we have a very happy mouse. Now, we also have choices about which AAV we use, and this is often very confusing. AAV is one type of virus, and it has many subtypes. There are actually virtually 100 of these, and you see the ones I have listed here that will target skeletal muscle. So if we put the gene in AAV 16589, it'll go to muscle. At Nationwide Children's Hospital, we choose to use AAV RH74. It goes to muscle, but also notice it goes to heart. And these two become our target organs for gene therapy. And we know from preclinical studies that it's been very effective in getting there. So the next question is it safe, and how can we be sure that it's safe? Well, in the preclinical studies in mice, we've injected it over 1,000 times, but the best safety studies are actually done in non-human primates because their genes are much more closely related to human genes. So we test the route of delivery here, and we've tested them in muscle in multiple ways and multiple doses in many monkeys. We tested to make sure it goes to the muscles that we want to by injecting it into the limb to get to the gastrocnemius muscle or the quadriceps muscle, two important muscle groups. And then we have injected it into the whole limb to make sure that it's safe. And finally, we've done systemic gene therapy studies that actually preceded what we do in the clinic. And this then becomes our package that we take to the FDA. And this is actually how things work. We took this to the FDA initially in 2006 when we did our first gene therapy trial for Duchenne. And this was and then later on, we modified this further. And this is the size of the package that we actually submit to the FDA. It's called IND, investigational new drug application. And this has to be supported by hundreds of pages of documentation before it'll be accepted. Once it's accepted by the FDA, we have to take it back to our own hospitals, and we have internal review boards that look clearly at the safety that we presented to the FDA and look at any ethical guidelines that are important. This has to do with the design of the trial. And we have another local group called the Institutional Biosafety Committee, which then provides NIH guidelines for doing any clinical trials. So we have a very good overview of clinical trials before we ever get to any patients. But when we do get to patients, we have to have patients sign a consent form and agree to the program. They have to understand clearly the gene therapy trial. Then if they do that and sign the consent form, as you see, this is essentially what we did. This family is now signing the consent form and being kind enough to take a picture for me. And then they have to pass eligibility requirements, normal blood tests, and normal physical examination. I'll show you that. But before, I want to point out one of the most important things, and often one of our biggest obstacles and confusion for patients in terms of the gene therapy. Patients can have pre-existing antibody to AAV. This is like if you were if you were exposed to COVID-19 and had never had any symptoms, your body would actually show that you had an antibody to AAV. If you have this antibody, it will, that we can check and must check before we get to the clinical trial. It requires a blood test, takes a few days to get back. And if you have this antibody, it will block the virus that's now carrying the gene, the microdystrophin gene. It will block it on its way to getting to heart and muscle. So the question then is, if that happens, what can we do about it? And we have about 10% of patients who have pre-existing antibody to AAV. We have potential ways of removing the antibody, and these are under study right now. We haven't done this in clinical trial. We have done this in the lab, remove the antibody by a filtering process called AAV. We've changed the serotype from RH74 to AAV9. We've used an endopeptidase, an enzyme that can degrade the antibody. There is an immunosuppressant cocktail, just like you would use for chemotherapy for cancer. And the last possibility is to change the vehicle for transfer. So you're no longer using AAV, but you're using a particle called a nanoparticle for gene transfer. So just a brief look at the blood tests we do. We make sure there's no kidney disease, diabetes, or liver disease in particular. These are the chemistries we use to exclude that. And you'll see in a minute exactly how that is done in the clinical trial. The next thing we must do is a muscle biopsy. And these are done pre-gene therapy and post-gene therapy to make sure the gene is working. This is an example of a picture that was done in a patient who was put to sleep before the clinical trial. And we marked the area in the posterior calf muscle that we're going to biopsy for the trial. And this is exactly how it's done in a real picture. We anesthetize the skin using an ultrasound probe. When we put in the needle into the muscle, this is the ultrasound probe that goes into the muscle, and then we can remove the muscle and send it to the lab for preclinical, and I'll show you a picture of that in a minute. The next thing we do is then we can, if all of these criteria are met, no antibody, no disease that would potentially lead to side effects, we have the virus now we can load into a syringe. This is in a solution, a saline solution that really will carry the virus. We load it. This is an actual picture from one of our gene therapy trials where we load it into an infusion pump just like this. We set up the infusion pump and then infuse it over an hour and 15 minutes. One of the things that often comes up during the trial is what happens during the trial. We carefully monitor. We have IVs in both extremities, as you see here. We monitor carefully during the trial heart rate, blood pressure, and EKG. And one of the most interesting things in the trial is we have no side effects during the trial. This is a totally blind process, and we're monitoring all the time during the process. The adverse events that we see during the trial after the day of gene delivery, usually starting about day 3, about 50% of the patients will develop nausea and vomiting or lose their appetite. We commonly see elevated liver enzymes, and sometimes we see platelet decrease. All these adverse events can be easily controlled and have never blocked progression in a clinical trial. So these are not anything to be feared and something we can control very easily. The next thing we do during the trial is we carefully test pre and post gene delivery, what we call the North Star ambulatory function. And the North Star measures multiple functions, actually 17 different measures. And we look at standing, we look at neck control, we look at getting up on one single step. And then in addition, we do time tests for 100-meter walk and climbing stairs and time to rise. Shut this thing off. And then we can move on to what happens after the trial. And this is one of the boys in our trial, like to show one. This is this kind of response is what we've seen. This boy is 18 months post gene therapy and essentially is normal. And that is the kind of success that we've had. Here's another one that shows a patient who is right at the time we injected the gene, going upstairs very carefully, not able to do a reciprocal stairs, goes up one at a time, and then 60 days post gene therapy here is able to climb stairs with ease. And these are two boys that were in the initial clinical trial that we have data now out to 2 years and looking very favorable for gene transfer. This is, I believe, my last slide where we show that this is analogous to the picture you saw earlier. No gene expression. This is the first 4 boys we did. No gene expression on their pre-muscle biopsy and robust gene expression. In this clinical trial, we had 81% of the muscle fibers showing dystrophin or microdystrophin expression. That's the largest amount that has ever been seen in a clinical trial. So everything looks very favorable for this. And we're moving on now to expanding the trial. I showed pictures of the first 4 boys. We're now nearly complete with another 40 patient trial. And we're going from the 40 patient trial to what we hope will be approval by the FDA in what we call a phase 3 multi-clinic, multi-center trial. So things are looking very favorable, and I'm happy to address any questions in the coffee break. Thank you very much.
Thank you so much, Dr. Mendell, for a beautiful, clear presentation, and really exciting to hear what has been happening and hearing of the history of what's been happening with gene therapy. And we really look forward to hearing more about this over the next few years. Thank you so much for doing that. So Rochelle, I'm going to hand back over to you at this point for you to explain what's going on now. Thank you.
Perfect. Thank you so much, everyone. That was an amazing session. We want to give you as much time as possible in those coffee breaks with Dr. Mendell and Dr. Campbell. So we are going to just push back the next session on innovative therapies. It's supposed to start at 1:30 Pacific and 4:30 Eastern. We're just going to push those back by about 10 minutes to allow for a full 20-minute coffee break. So we will end this session now. Feel free to go into the coffee break, have a good 20-minute chat with Dr. Campbell and Dr. Mendell. I'm sure they'll stay on past that if you'd like. And we'll come back and start the next session, keynote session on innovative therapies in approximately 20 minutes. Thank you so much, everyone. Bye-bye.